Hello. My presentation is on coffee consumption is associated with intestinal loss in Bacter, Saccharolyticus abundance and prevalence across multiple cohorts. So why should we care about this field? So coffee is one of the most widely consumed beverages in the world. However, there are some specific effects on the human gut microbiome that coffee has that is not largely defined. This study asked, does coffee consistently result in substantial effects on the bacteria, Bacter, as Saccharolyticus, and other areas in the human microbiome? Researchers found that this bacteria is robustly and specifically enriched in coffee drinkers. This is important because it allows it to give it a unique advantage, I would say, to this bacterium because it has unique metabolic pathways. Some background. So before this study, we knew that the diet has some kind of effect in the gut microbiome, but a lot of the evidence wasn't very consistent and not very species specific. Um, I know there was a few other studies that looked at the coffee's effects on the microbiome, and none of them made a specific link between a specific coffee compound and a specific microbe. This leaves us with a gap of knowledge of what we know and how coffee directly affects us. So quinic acid is a bioactive compound that is abundant in coffee. And people that regularly consume coffee have high amounts of this in their gut. This bacteria is a common microbe that is in the Ruminococcaea family. This microbe is a saccharolytic, which means it does not use carbohydrates for energy. And this is very important because since this bacteria doesn't use carbohydrates for energy, it is able to use other sources of energy, such as quinic acid. Connection. So since this bacteria doesn't use carbohydrates, like I said, it's able to use different sources of quinic acid. And since this bacteria is able to use quinic acid, it uses it as selective growth advantage, which in return allows it to grow more. Since the other bacteria's the most common source is carbohydrates, then it allows this bacteria to grow more since they're abundant in quinic acid in the gut, it allows it to grow more since the energy source is abundant. And like I said, it also gives other microbes the ability to take their energy from a non-common source, and it helps it to grow more effectively. Urgency. So it's very important to learn the microbiome's response to dietary factors such as coffee, since the microbiome is very important part of our health, such as inflammation, metabolic disease, and immune function. Coffee is the second most consumed beverage in the world. There's about 1 billion coffee drinkers. So I think it's very important to understand the positive microbiome implications to have so we can have specific health nutrition goals and microbiome targeted therapies. So I feel like the urgency comes from the need to collect more data and how our gut microbiome reacts to the food that we intake. You can also see which like food is best to eat as more microbiome research comes out and we can understand more about that. So many studies have shown that coffee is linked to lowering some chronic illnesses and various cancers. And the important thing is that researchers are now trying to find out what specifically makes that happen. Um, and I feel with more research, this will be able to spark more interest in this area in science, so we can learn more about it, specifically in coffee. So this article, so this study aims to answer which microorganisms are most closely associated with regular coffee consumers and like what mechanism creates this link. So like their hypothesis was that specific compounds in coffee, especially quinic acid, like selectively stimulates the growth of Lawbacter acroleticus, and this is a rare butyrate producing bacterium. So methodology. So in this study, there was about 23,000 individuals, and it came from five cohorts and from the US and the UK. So coffee consumption was self-reported, and it was grouped between non-consumers, moderate consumers, and high consumers of coffee. So the researchers analyzed different things such as different lifestyles, metabolic syndromes, and plasma metabolics in the participants. And they chose the shotgun metagenomic sequencing in order to like identify the specific species and strains in the microbe. And one of the good things about using this was that it allowed them to be able to only see like the microbes that consist of DNA and not other microbes such as viruses. Using this style also allowed the researchers to collect data from various regions in the world, diets, and health statuses. Part two. So participants of the study were separated, like I said, in three groups, and their fecal samples were collected, and this was to ensure that the shotgun sequencing was allowed the researchers to study the specific species composition. They also controlled the statistic samples by varying factors such as age, sex, BMI, and fiber intake. Part three. So, in this study, they used in vitro experiments, and the L. acroleticus was cultured with coffee extracts and purified quinic acid to test whether these compounds directly promoted its growth. They also compared these results to the same control but in microbes such as E. coli. Also, the researchers used like they also cultured L. acroleticus with decaf coffee and a control group, and this was just to ensure that the results were accurate. And then the plasma metabolomics. Um, there is a subset of around 438 participants, and this is to identify the coffee metabolics. So this was like to determine like what is the linking microbial abundance. Some definitions that were important was butyrate, and this is a short fatty acid that provides the body with anti-inflammatory support and colonic health aid. Quinic acid is a polyphenol that is abundant in coffee and can reach the gut microbiome. Cross-feeding. So this is the ability of one microbe to create metabolites that can be used as nutrients for other microbes. The results. So the first part of the results was based on the that this study concluded that there was a strong connection with drinking coffee and the L. acroleticus. It was found that this bacteria was mostly consistent, um, most abundant in the coffee consumers. And this finding was consistent across all five cohorts. And this was the only bacteria that was found in all five. There were other microbes that were found to increase when there was a high percentage of coffee intake. Um, these were the Bacillobacterium prausitzizi and Oscilliospiraceae, which are also butyrate producers. There were also some microbes that were found to have a decrease, such as the Erysipelotrichaceae and some Firmicutes and Bacteroidetes. Um, and like I said, the only bacteria that was found in all five cohorts was the L. acidolyticus. Part two. So scientists were able to create co-occurrence networks, and this revealed that the bacteria formed a functional group with other fiber fermenting butyrate producing bacteria. And models could predict the coffee consumption with around 93% accuracy. Um, and then they also found that this bacteria was the single most informative marker for coffee intake. They also discussed how like these fatty acids promote immune support, colon health, and defense for disease that cause their erythropoietic responses. So the metabolic pathways. So on the diagram on the left, it shows that the metabolic flow of non-digestible carbohydrates or fibers that reach the colon. These are fermented by microbes into various intermediates like lactate, pyruvate, acetate, formate, depending on the species. And is and these intermediates are then used by other microbes in a process called cross-feeding. For example, in butyrogenic cross-feeding pathway, the lactate and acetate are converted into butyrate. And this is important because it's a short chain fatty acid that supports gut barrier and reduces inflammation and has like it also helps protect against colon cancer. There are also acetogenic and propionate pathways, and they result in other metabolites. But in this study, we're most interested in the butyrate pathway, since the Lawson-Bacter ac acidolyticus um, is the one that was discussed in this study. And like I said before, there's also other bacteria that are known as butyrate producers, such as the Bacillobacterium and the Roseberia. And to the diagram on the right, it shows the classic model and the Pateron model. And in the classic model, we have two types of microbes. The first one's a primary degrader, like Bacteroides um, beta-domigron. And these break down complex fibers and release intermediate metabolites like acetate. Then we also have secondary resource specialists, such as the Phycobacterium procedens that use acetate to produce butyrate. Also, in this framework, the microbes act somewhat independently by exchanging a few byproducts, and the host benefits downstream from the production of butyrate. But in the next picture, in the Panteron model, so this is a little more complex and communal, since there is an extensive exchange of metabolites and extracellular electron transfers, and there's like um a large web of cross feeding that benefits like not just one species but the entire ecosystem. And this is important since since this study focuses on the Lawson-Bacter asac asacrolyticus. So this dense network creates a metabolically rich environment, and essentially it's almost like a microbial pantry. And the presence of the coffee derived compounds like the quinic acid, it gives a certain microbes a growth edge. And this allows the butyrate producers an environment where they can grow more rapidly and have selective growth. Part three. So in the subset of fraudulent members, this was to determine the coffee compound located in the blood. And researchers used in vitro to see if those compounds or coffee were stimulating the microbial growth. Um, and then in the study, it showed that the L acetolyticus grew 3.5 times more in media supplemented with coffee extract or quinic acid. And then in the control groups, it showed that it had no response. So this shows how there's like a direct evidence that entails that coffee enriches L-acetolyticus by quinic acid and these observational findings. Part three. So the findings in the plasma metabolomics showed that there's, like I said, a strong correlation. And this was only present in the coffee induced cultures. It showed that there were other acids present, but quinic acid was definitely the most robust. Um, and it also showed, like I said, like since it was the most robust, it showed that it was the key substrate in the study. And then the control bacteria such as E. coli showed no stimulation in growth, which confirmed the hypothesis. Discussion. So why is this important? So coffee consumption like shapes how like the gut microbiome can like be like precise and predictable in a way that we can like predict what will happen when someone drinks a lot of coffee. And these can determine like our health outcomes. Like I said before, that there's over 1 billion coffee drinkers in the world. So I think it's really important to understand how this beverage can shape our microbiome. And this connection between the L-acetolyticus and the quinic acid shows there's a direct link between these two from a compound to a bacteria. And it shows how big of an impact it can have on our gut microbiome and dietary factors. Broader implications. So it did show that there's potential for developing microbiome targeted functional foods. I feel like this also like can encourage people to like watch more on what they eat and like um, learn about how their food affects their body. And I pretty much think it just adds more evidence that there's a direct link between diets and microbial and systematic health. So some strengths in the study showed that, well, one thing was that it was a huge sample size with 23,000 people, and it was across different geological locations. It also was able to be viewed from different pathways, which I thought was very interesting. Some limitations in the study showed how um, like in vitro systems, like it can not fully replicate the gut environment. So that's definitely something that may not give like full accuracy. Also, the study didn't really focus how much like it would affect someone in the long term. Basically, it was more just short term. Future directions. I feel like there could be like kind of clinical trials with kind acid, or L-saccharolyticus supplements. I also think that, like I said, like there should be more research on the long-term health effects of the microbial shifts from coffee. My personal takeaway from this, I feel like this study demonstrates like the growth of the microbiome science to study to represent how our everyday choices and like the food we eat every day and like our habits are able to shape our health in such a big way and shape our biology. I feel like this is also a good way to like spark interest and spark more research in the future on not just coffee, but maybe other common foods and beverages. Some questions that can be asked are how could genetics play a role in the influence of microbes in the gut towards a response to coffee? What could be some negative outcomes of an influx of L. saccharolyticus? Are other dietary sources that contain an abundance of quinic acid able to replicate the same response to the microbiome? Can coffee be used as a targeted prebiotic? Would an in vivo supplementation mimic the same findings, or would there have been a different outcome? And in conclusion, I believe that this concept that there's a direct effect and human microbiome is really huge. And like I said, I feel like it could spark more research being done. And like I said, like it can in the future maybe be even used as a selective prebiotic. I feel like also this research helps to understand that the gut microbiome is not um is able to be affected by what we eat and be able to predict how our health will be. And I feel like this is very important to be able to understand how like if we eat this amount of food or eat this every day, like what the effects will be in our body, um and how like our health will be affected by that. And these are my references.